Hello students, I'm Wikim Siliang. I'll be teaching biology today. The chapter that I'm going to teach today is the molecular basis of inheritance. Now in this chapter we're going to discuss about the structure of DNA, genetic material that prove the experiment that proved the genetic material, replications, transcriptions, translation, DNA fingerprinting, and lag operand. Before we start up with the chapter, let me first explain the introduction of this chapter. That is, let's try to understand the meaning of genetic material. What this genetic material is? Something that has been transferred from the parents to the offspring. For example, like the color of the hair, the height of the organism, and many more. So, what character had been transferred from the parents to the offspring? That is considered as a genetic material. But there are some criteria that to consider a genetic material, a molecule should possess some character. So the first character is, point number 1, they should be able to replicate. Able to replicate. It means when a cell divides, they should be able to multiply. The second point is their structure should be stable chemically and structurally. They should be stable. The genetic material should be stable chemically and structurally. Stable chemically and structurally. Point number 3, they should provide some slow changes. Slow changes in the sense like mutation. You know that mutation is harmful, but slow mutations are useful in the process of, in the preparation or in the manufacture of protein in our living system. So slow mutations, the genetic material, they should provide slow mutation rather than fast mutation. Should provide slow mutation. Because slow mutations, why they are necessary? Because these are required for the process of evolution. The next 4 important thing is that they should be able to translate by the process of transcription. I'm going to discuss detail that one later on. I'll just write a point right now. Should able to translate by the process of transcription. What is transcription? It's nothing but if the DNA get converted to RNA, that is called transcription. We're going to go detail on this, but these are the 4 important characters that a material should possess. That is, they should be able to replicate, they should provide stability chemically and structurally, they should provide slow mutation because that is required for the process of evolution. The 4th thing is that they should be able to translate the process of transcription, that is DNA to RNA. Now the next thing that we have to know is about the genetic material. The types of genetic material. In class 11th or class 10th, you have already studied about the types of genetic material, which you have known, and we call that as nucleic acid. Nucleic acid. You know that there are 2 types of nucleic acid, that is DNA and RNA. DNA is deoxyribonucleic acid. And RNA is ribonucleic acid. Now, these are the two types of nucleic acid. One of the important things that I want to tell you is that scientists have discovered that RNA is the first genetic material. First genetic material. RNA is the first genetic material. Since we are talking about the genetic material, the most stable one is our DNA. I'm going to prove why is it DNA than the RNA. Now, this DNA was first observed by Frederick Mesher in 1869 in pus cells. First observed by Frederick Mesher in 1869 in pus cells. And in other books, in higher books, you will find that in salmon sperms. So the scientist Frederick Mesher had observed this DNA as a genetic material in 1869 in pus cells, but he named it as nuclein. Frederick Mesher, he named it as nuclein. Later on, in 1889, Altman, a scientist named as Altman, gave the term nucleic acid. So this is about the history of those scientists who have discovered and how later on how the Altman has given the term nucleic acid. Now let's go with the detailed introductions of the two types of nucleic acid, their composition and their structure. Composition of nucleic acid. It can be DNA or RNA, but they are composed of three important parts. That is, the first one is pentose sugar. The first composition is pentose sugar. Now, pentose sugar is of two types. It is ribose and deoxyribose. This ribose sugar you will find in RNA and deoxyribose for DNA. So specifically, both are different for DNA and RNA. So, but when we say pentose, what does pentose mean? There will be 5 carbon atoms. I'll show you the diagram. Please, I want all the students to look this structural composition properly, because many of the time in the competitive examinations, this type of questions are asked. So, let me make the structure. is CH2OH. I'll represent the carbon. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So the name itself showing 5. 5 means pentose, sugar. This is for the ribose. Let's make for deoxyribose. Right here, CH2OH and 1, 2, 3, 4. So here I can just write as 1H. So if you see the chemical structure, you can see that both ribose and deoxyribose, the only difference is in the carbon number 2, which doesn't have hydroxyl group, OH group. But here in the ribose, you can see that there are 2 hydroxyl group. So this is one of the difference between DNA and RNA. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. And that is the reason that RNA become more reactive also. Why? Because of the presence of these 2 hydroxyl group. In other way, we can say that the more the number of hydroxyl group, they become the reactive group. So the first important thing is your pentose sugar. Next, nitrogenous base. The second most important composition is nitrogenous base. It's of 2 types, that is purine and pyrimidine. So this purine is of 2 types again, that is adenine and guanine. So you can represent by A and G, whereas pyrimidine are cytosine, thymine, another one is uracil. So there will be 3 pyrimidine. That will be different for the case of RNA and DNA, and for purine we have adenine and guanine. Let's try to understand what this purine and pyrimidine means in detail. Now, usually purines are 2-membered ring. 2-membered ring, or I should say dicyclic. Okay. Whereas pyrimidine is 1-membered ring. 1-membered ring compounds, or I should say monocyclic. The next important point that you have to remember is, let's write this. Point number 3, the purine is, there are 4 nitrogen atoms. 4 nitrogen atoms. And these 4 nitrogen atoms are located at first carbon, the 3rd carbon, the 7th carbon and the 9th carbon. Okay. First, I see the first position, the second, the first, 3rd, fifth and 7th and 9th position. It means that the 4 nitrogen atoms are located at the 1st, 3rd, 7th and 9th position. Whereas in pyrimidine, there will be only 2 nitrogen atoms. And these 2 nitrogen atoms are located at 1st and 3rd position. So let me show you the structure of this purine. Now this purine, I said that these are 2-membered ring. So I will not make the exact structure of that, but an idea of this 2-membered ring structure. So in this way you have to give the position. The position should start from 1. So when I say first position, so here is the position where you get your nitrogen. When I say third, this is the position that you are getting your nitrogen. 7th, this is the position where you are getting your nitrogen atom, and this is the position where you are getting your nitrogen atom. Whereas in the case of pyrimidine, I said it should be only 1-membered ring compound. So I said it should be only 6 carbon. So the first carbon will be here, 1 nitrogen, and in the 3rd carbon. So the structure of purine and pyrimidine is very important for the point of competitive examination also. So please, I want all of you to make the structure that you have studied in class 11, okay, in the chapter of biomolecules, where the structure of adenine, guanine, cytosine, uracil and thymine. Please try to make the structure and memorize the chemical structure of this purine and pyrimidine. The most important point is number 4, the first carbon of the pentose sugar binds with or attach with, for the purine, okay, for the purine you should attach at the 9th position. So this is the 9th position, okay. The pentose sugar should bind at the 9th position. Whereas in the case of the pyrimidine, the first carbon of the pentose sugar binds with 3rd position, okay, 3rd position. The first carbon of the pentose sugar should bind at the 3rd position of the nitrogen atom. I'll just write as nitrogen atom, nitrogen atom. So these are the few important points between the purine and pyrimidine. Let me revise it. Purine, as I said, we have adenine and guanine, 2-membered ring compounds. Pyrimidine, we have uracil, cytosine, thymine, 1-membered ring compounds. You can see only 1 ring structure. And there are 4 nitrogen atoms at the 1st, 3rd, 7th and 9th position. For pyrimidine, it is in the 1st and the 3rd position. And the last most important point is the first carbon of the pentose sugar. The first carbon of the pentose sugar binds at the 9th position of your nitrogen atom, and the first carbon of the pentose sugar binds at the 3rd position of the nitrogen atom. So these are the differences between the purine and pyrimidine. Now, the first one that we have discussed is pentose. The first one that we discussed was pentose sugar. Next, we discuss on nitrogenous bases. The third important component is phosphoric acid. Phosphoric acid. I'll make the structure of this. There will be 3 hydroxyl group. So these are the 3 important components of your nucleic acid. When I say nucleic acid, nucleic acid is nothing but the first one is pentose sugar. Next is nitrogenous base. The last one is phosphoric acid. So in the next class, we'll discuss the term called as nucleotide and nucleoside in order to understand the structure of DNA. Thank you.